Hello, students of Anatomy and Physiology 2. I would like to give my first lecture today. We'll see. We hope that this works out well for you and that you can profit from it. My lecture today is actually on the small intestine. The small intestine has two functions. One is digestion of food and the other one is absorption of nutrients. I want to first focus on the digestion of food. Now you know that of course food is taken in through the mouth and digestion starts in the mouth. So the amylase in the mouth and your teeth, they are already preparing the food for further breakdown. In the stomach then pepsin is waiting together with hydrochloric acid to break down the food even more. And after the food has gone through the digestion of pepsin and the gastric lipase, you get a paste or paste-like fluid that is called chyme. This chyme enters the first part of the small intestine, which is called the duodenum, through the pyloric sphincter. And in the duodenum, now much more digestion is taking place. There are two sets of enzymes that start to work on the food. The first set comes from the pancreas, which you, I hope, remember that the pancreas is an exocrine gland and produces digestive juices. These digestive juices are being released into the duodenum. So the pancreatic juices contain a whole mixture of enzymes. And these enzymes basically work on all four types of macromolecules that you hopefully have read about. So there are the following pancreatic enzymes that are starting to work on this food. The first one, so these are enzymes from the pancreas, and they are being released into the duodenum. First of all, trypsin and chymotrypsin are enzymes that break down proteins or short pieces of proteins, which are called peptides. These enzymes, trypsin and chymotrypsin, break these proteins or peptides down to amino acids. Then we have pancreatic lipase. The word lipase tells you already that this enzyme breaks down fats into fatty acids and glycerol. Then we have pancreatic amylase. Pancreatic amylase, just like salivary amylase, breaks down starch or short sugars into either disaccharides or monosaccharides. And finally, the last enzyme is a nuclease, or actually a whole group of nucleases that break down nucleic acids such as DNA and RNA. So we have basically enzymes for all four macromolecules. We have enzymes that break down proteins, enzymes that break down fats, enzymes that break down carbohydrates, and enzymes that break down nucleic acids. These enzymes are being released into the duodenum and work inside the duodenum. At the same time, the duodenum has its own set of enzymes that work on the food. I want to turn this quickly around. So now we are going to talk about the enzymes that are already located in the duodenum. They are so-called intestinal enzymes. So we have here enzymes located inside the duodenum. These enzymes are called usually intestinal. So we have intestinal lipase. Again, lipase being an enzyme that breaks down fats. Then we have a group of enzymes called sucrase, lactase, and maltase. These enzymes break down short sugars, or also called disaccharides. So we want to note that they break down disaccharides. Disaccharides are still too big to pass through the wall of the small intestine to get into the blood. So they need to be broken down into monosaccharides. And finally, we have a third enzyme, and that is the peptidase. Peptidase breaks down peptides, and peptides being short pieces of proteins. Remember, when you eat, for example, egg or some protein-rich food like meat, these proteins get pre-digested already in the stomach through the enzyme pepsin. Now you remember that proteins are very, very long and big molecules, and pepsin cannot break these long molecules completely down into amino acids. Usually what happens that a long protein like this one gets broken down into so-called peptides that then by the peptidase get finally broken down into many, many amino acids. These amino acids finally are small enough to pass through the wall of the small intestine into the bloodstream. Remember, the whole point for us to eat is to bring building materials into our body, but the food we eat, these molecules are way too big for us to absorb them. So these proteins are like scissors that basically break down these huge molecules that we find in food into tiny little micromolecules that can be absorbed in the small intestine. So we have talked about the enzymes that come from the pancreas and the enzymes that are already located in the duodenum. By the way, these enzymes that are located in the duodenum have a special name, and this name is brush border enzymes. So I'm going to write this down here, brush border enzymes. The reason for this name is that these enzymes are located in the intestinal wall. They are located in cells that make up the lining of the small intestine. So these enzymes are not floating around inside the small intestine, but they are basically stationary, and the food, as it's being pushed through by peristaltis, gets exposed to these enzymes. And these enzymes then work on the food, and as the food breaks down, the food gets immediately absorbed. That takes me actually to the second part. Oh wait, I forgot one thing, because we talked about the pancreas, we talked the intestinal enzymes. Now there's one more thing that gets added to the mix, and that is also very important, and that is actually bile. Now bile is originally being made by the liver. The liver produces an abundance of bile, and therefore the bile gets stored in the so-called gallbladder. So the gallbladder is a storage place for the bile, but it's not being made there. The bile usually is only released when chyme enters the duodenum. So that is why we need a storage place, because the liver constantly is making bile, and bile should not be constantly be delivered to the duodenum, only when it's really needed. So let me explain to you why we need bile. Bile breaks down fat droplets. Let me draw a fat droplet here. Let's say you ate some bacon, or you had some butter, or some whipping cream. You know there is a lot of fat in these nutrients. And as these fats, even though lipase in the stomach already worked on this a little bit, we still have bigger fat droplets. And the lipases from the pancreas and the intestinal lipase now try to break down these huge fat droplets. Now when bile gets delivered through the bile duct into the duodenum, what happens is that actually these huge droplets break down into smaller droplets. And you see, and I'm just trying to kind of sketch this here real quick. You see that these big fat droplets get dissolved into smaller droplets. Now you remember possibly our experiment with chewing, where we learned that smaller pieces get processed much quicker, for example, when they get dissolved or when they have to react chemically. That's the same thing with these small droplets here. The bile produces these small droplets, and now lipase can actually get to these droplets from all sides. And that makes the breakdown of these fats into fatty acids and glycerol much easier. So a person who does not produce a lot of bile, they have a much harder time to digest fats. So you can see how this is actually a very useful process in fat digestion. Okay, we call this process of breaking down big molecules, big fat droplets into smaller droplets, emulsification. That is the name. Emulsification is the process that breaks down big droplets into smaller droplets. Now I want to let you know that breakdown does not mean that the fats get broken down chemically. Fats are still fats, but just like you find in your soup those big droplets of fat swimming and floating on top, imagine that gets changed just to smaller tiny droplets. But the fats itself, the molecules, are still intact. That means the lipase still has to break these fats down into glycerol and fatty acids. So I want to stop the video here now and want to then later publish a second video which deals with the absorption of the nutrients in the small intestine. Thank you.